Hello everyone, my name's Belinda O'Rourke and I'm a Technical Officer in the Microbiology Lab at Elizabeth MacArthur Agricultural Institute. Today I'm here to talk to you a little bit about bee diseases and the laboratory diagnostics that we work on. The Elizabeth MacArthur Agricultural Institute was established in 1990 and it's situated near Campbelltown in New South Wales, and it's a world-renowned premier biosecurity facility and enhances food and fibre production and helps protect the environment. It's run by the New South Wales Government, by the New South Wales Department of Planning, Industry and Environment, and its focus is on research, veterinary diagnostics, plant health, community education and farming. And we have a number of labs on site, including virology, serology, parasitology, microbiology, genetics, plant and soil health laboratories, and importantly for today, we have two labs working on bee disease diagnostics and research, and they are the virology and microbiology labs. Today, I'm going to be mostly focusing on the work in microbiology laboratory, which is the laboratory that I work in. And mainly we are a veterinary pathology diagnostics testing lab, and we usually test livestock and bees. But we do also get wildlife and domestic animals in, as well as exotic animals from zoos. So we have quite a range of tests that we run. We have a team of 8, plus we have support staff. And our bee work includes microscopy for AFB and EFB detection, Nasima detection, we do honey culture for AFB, we do OTC testing, and we have a small section running some research on our bee work. Here's a quick look back at history, and the first records of beekeeping were about 9,000 years ago, and bees were housed in pottery vessels. This picture here is of Egyptian art showing beekeeping about 4,500 years ago, and I think that's pretty cool. So if we jump forward a couple of thousand years to Aristotle, he was the first to describe diseases and disorders in honeybees, and his description was a lassitude on the part of bees and a malodorous of the hive, and that just means weak, lazy bees and a foul smell of the hive. And although the descriptions could not identify the disorders with any great certainty, it was the first time that disease was described in honeybees. If we jump forward again to 1771, honeybee brood disease was described by Adam Chirac, and he characterised it by a foul smell, and he was the man who coined the term foul brood. Another 100 years later, and it was finally determined that there were 2 different types of foul brood. One was mild and curable, and one was malignant and incurable, and both were called foul brood. It was in 1906 that G. White first identified Bacillus larvae, which is now been renamed as Bacillus larvae, as the cause of American foul brood. And it was at this stage that AFB and EFB were formally recognised as separate diseases. But it wasn't until 1919 that he discovered that EFB was caused by Bacillus pluton, which is now called Melissococcus plutonius, and it was found in many European countries and in America. It was in this era, in the early 1900s, that other diseases, such as Nasima and viral diseases of honeybees, were beginning to be identified. And today, bees are known to suffer from over 30 infectious diseases and parasites. Now, to bring it home a little bit closer to us, this is the timeline of detection of some of the diseases and pests in honeybees in New South Wales. So in 1976, AFB and EFB were detected in New South Wales. In 1992, chalk brood and Nasima apis were detected. In 2002, small hive beetle was detected. And in 2007, Nasima ceranae was detected in New South Wales. Now, these are the bacteria and pests and other diseases that we work on in the microbiology laboratory, but between 1987 and 2015, a whole host of viral bee diseases was also detected. So I'm just going to focus in a little bit on a couple of diseases that we do a lot of diagnostic work on for New South Wales and other states and territories. AFB is a fatal disease of honeybee brood. the larvae in the pupae. It usually kills beehives, and it's caused by Paenobacillus larvae. Unfortunately, it is the most destructive and widespread of the honeybee brood diseases. AFB infection is caused by the spores of the bacteria, and most of those spores are removed from the hive by adult bees. It is, however, the youngest of the larvae that are most susceptible to infection by the spores, and it only takes about 10 spores to infect a larvae that's less than 24 hours old. As they age, they require more spores to infect them, so a larvae 24 to 36 hours old requires 500 to 1,000 spores, and once they're 48 hours or more old, it takes millions of spores to infect them. So really it's those less than 24-hour-old larvae that are most susceptible to infection by the spores and therefore catching the AFB disease. And there's a couple of ways that AFB can spread between hives, and it's often an interchange of contaminated equipment between infected hives and non-infected hives. Can occur by bees robbing honey from diseased hives and taking it back to healthy hives, and there's a low risk from drifting bees. And unfortunately, there's no treatment for American foul brood, and the only options are eradication of the whole system, which is destruction of the hives by burning or by gamma radiation. Here we have examples of healthy brood on the left and diseased brood comb on the right. When you're looking for disease in the comb, you're looking for irregular brood patterns, a mixture of capped and uncapped cells. Individual cells might be sunken, darker in colour, greasy looking. There could be perforations in the caps as well. And you can see on the right here on the picture in the middle, we have the dried out scale in here at the bottom of the cells, and that's at later stages of infection. And then down the bottom, on the bottom slide, we have the brown goopy ropiness test that we look for in AFB. And this is the sample that you would take and send in to us on a smear, or you can collect larvae, dead larvae, and spread them on a smear as well. Moving on to EFB, or European foul brood, it is also a brood disease which usually is not fatal. It is widespread and is endemic to all states and territories of the eastern of Australia. It has not yet been discovered in West Australia. It's caused by the bacterium Melissococcus plutoneus, and it has a secondary invader caused by a bacteria Paenobacillus alvei, not to be confused with Paenobacillus larvae, which is the AFB bacteria. So here we have two examples of brood comb that's infected with European foul brood, or Melissococcus plutoneus and Paenobacillus alvei. It's characterized by patchy brood comb, uncapped brood cells, and the dead and dying larvae will often have this brownish yellowish tinge to it, which you can see in this slide here with this and this larvae both having that tinge to it. Once the larvae die, and they also become molten or brown and gloopy, and there's a distinct smell with European foul brood, and often when it comes into the lab we can smell that before we can see anything. The incidence of EFB is generally higher when the hive is under stress, and that can be caused by climate conditions, excessive rain, cool climate, movement of the hive, poor nutrition, and heavy infections of EFB will infect a large percentage of the brood comb, a weakening of the colony over time, and possibly leading to the death of the colony. However, it's not as fatal as AFB. Now, treatment of European foul brood, there is an option, and that is treatment with oxytetracycline, and it is a prescription antibiotic, but it is the only treatment approved in Australia at this time. So a discussion with DPI on treatment of that is always recommended. So Nocemosis is a disease of adult honeybees, and it's a silent killer of them. It's caused by microsporidium, which are tiny parasites that live in the gut of the adult bees, and the two that are identified are Nasima apis and Nasima ceranae. And identifying it in the hive is difficult, but in general, infected bees live only about half as long as non-infected bees. Queens are superseded within 2 to 8 weeks of becoming infected. It usually occurs in late winter, early spring. You'll notice a dwindling number of bees and a decrease in honey production, decrease in brood production due to the queen being less productive, and it can kill off hives. Chalk brood is another disease that we look for in the lab, and it's the only significant fungal disease of honeybee brood, and it's caused by Ascosphaera apis. And identification is via the brood comb. So if you're going to send something to us, it's brood comb that we need to see with the mummies, and that is the larvae that are covered in the fungal growth, which is down here in these ones here. And later they dry out and become grey or white or black chalky, which is this one here. So these are what we call mummies, and that's what we're looking for when you send them in to us. So as I said, testing in our microbiology lab, it includes microscopy when we're looking for the spores of Pinobacillus larvae. We're looking for Melissococcus plutoneus down the microscope, the spores of Pinobacillus alvei, which is the secondary invader of EFB. We do microscopy for Nasima species. We do culture on honey for Pinobacillus larvae. We do check comb for chalk brood. We also have developed testing for the oxytetracycline in hive products. So we have a test for that now. And like I said, we have some research on the go, which my colleague Kushbu will be talking about later. Now, this is straight from our website, the State Veterinary Diagnostics Laboratory website. And on here you can see that these are the samples that we require to run our testing. And on here we can see that for the honey we've got a minimum of 100 mils. The website at the moment states 75 mils, but we are updating that to be 100 mils, and you'll see in a moment why we're doing that. So I just wanted to give you a quick look at what it is we actually do with your samples once we get them, and how we do identify the different diseases, mostly focusing on AFB and EFB and a little bit on Nasima. So these slides you can see in this picture are what are sent to us, and they have the larvae on the slide, so you can put up to 3 larvae on each slide, smush them on and package it up carefully. If you've got that ropiness, or you've taken some sample from a brood comb, you can send it in on any hard surface and we will sort out making a slide from that, preferably slides, but if they're too hard to get a hold of, you can always send it in on anything. And we've had mirrors, we've had coffee cup lids, we've had all sorts of submissions sent in to us, and we will always try and come up with an answer as best we can. So what we do with those slides is we will heat fix the sample to the slide, and then we will stain it with this pink stain here, and that just brings up the spores and bacteria for us to be able to identify. And this is one of our ex-colleagues. This is in the lab, and he's looking at some bee slides under the microscope. And the picture on the right is generally what you can see. It's about the size that we come across. And in the next slide, you can see here that these little spores here are the ones that we're looking for. And there's thousands of them, millions of them. And when we identify these, it's AFB positive. In this page, you can see EFB. Now we treat the slides the same, we stain them up, we look at them under the microscope at 1,000 times, and you can see here that we have the Melissococcus plutoneus in this slide. And they're tiny little lemon-shaped circles that we can see. and that is EFB positive too. On the right slide, you can see we have lots of spores. Now these spores here, these are the Paenobacillus alvei, or the secondary invader of European foulbrood. In this slide here, we can also just see up here, and in a few different spots, are some leftover Melissococcus plutonius. So those two slides there become EFB positive. So when we requested to culture for AFB, or Paenobacillus larvae, what we're looking for are the live spores in a sample. And we do this by running it through a process, and we selectively culture on special plates which knock out other bacteria that would stop us from identifying the Paenobacillus larvae. And when you send us the sample, if your sample turns out to be positive, your results will come out with an explanation on the percentage likelihood of AFB disease in one or more of the hives that honey was collected for. Now this percentage is result, and it's been determined from research when developing the procedure, and that's the reason why we request the minimum quantity to run it, because our test has been developed with using 75 mils of honey. So if we have less than that, our results don't mean it quite as much and aren't validated. And that's why we request 100 mils, so we've got plenty to use. And this is one of our current lab staff running our honey culture procedure. And what we do is we get our honey in that you guys send us, and we warm it up till it's runny, and then we mix it in these bottles up here with a buffer liquid, and we shake it to emulsify it, and then we spin that for a long time, and we spin it down so that all the spores collect in the very bottom of these tubes here, and from that we then heat that sample that's right down the bottom that's hopefully got either no spores or all the spores from that sample in the bottom of that. We heat it up and we place it onto these plates up the back here, and they are our selective plates which help us knock out other bacteria and help grow the Paenobacillus larvae. And this plate up the top here is an example of a positive growth for Paenobacillus larvae. Now this is a heavy growth. This is a control organism that we have in the lab that we use to validate our tests and our media that we make, the plates here. And that's what it looks like. And colony morphology, what the bacteria actually looks like, and some other factors are what we use to identify the Paenobacillus larvae. And in the last few years, we have acquired a MALDI-TOF machine, which is the picture in the bottom right here. And that machine identifies bacteria. So we place a sample off the culture plate into the MALDI-TOF machine, and that breaks it up into proteins and gives us a profile and gives us an identification of Paenobacillus larvae or whatever other bacteria we may have cultured. And it just gives us additional confidence in a positive result. This page here is our Nasima testing procedure. So for Nasima testing, we need 10 to 25 adult bees. Now you can send them into us like this one in a container. They're already dead. Or we like them also preserved in some alcohol. The adult bees are ground up in the machine up the top here, and this second picture. And what we're trying to do is get the Nasima parasite out of the gut of the bee. So we crunch it up and we place a tiny amount, only 3 microliters onto a slide, and then we look at that under the microscope as well. And those parasites, like you can see in this picture, they light up under dark field microscopy, so we can identify them through that. And here I just wanted to give everyone a bit of an idea on how to get your samples to us. I know it can be a bit tricky to find on the website, so I've included the link up the top here for anybody who needs it, or you can just search in your search bar for the State Veterinary Diagnostics Laboratories, and that will bring up the Department of Primary Industries here, and you can click on that page. If you click on this test list pricing over here, that will take you to a page over here. And that second page up here is where you can click on all the different tests. But as beekeepers and honey producers, if we click on one of these three here, the BMIC, American Foul Brood and European Foul Brood Microscopy, or the culture for the honey, if you click on those, it will bring you to another page. And then from that, you can find this further identification or further information sheet down the bottom here, and that will lead you to this form. Now this is the bee diagnostics form. If you fill that in and send it to us, that gives us as much background as you can. It gives us an idea of what is happening in your hives or with your bees, and we can have an idea of what we're looking for. Any further information is on the next slide. And on this one, it's the same process, but this one gives you a guide for collecting samples to send in to us. So you can get a DPI prime fact sheet, which helps to show you how to collect samples to send in to us. So it's the same procedure as getting into the submission form that we send in to us. You click on the test list and pricing, and then one of the three B tests, and you can go down and you get to specimen requirements, and you can click on how to collect samples and prepare samples, and that takes you to the prime facts sheet. So they're all helpful tools in order to get your sample to us. If filling in an official form is impractical because you're out in the field, or you can't print it off, or it's just not working, handwrite us a form, give us the information, give us some background. It all helps in our diagnosis of your bees. And this is, I just wanted to talk through a few things that come into the lab when we're trying to run our diagnosis on them. And you can see in this left picture, we've got three different types of samples that get submitted to us. We've got your honey, we've got your smears, and then the bees. These are very good examples of sending us samples, and we will take them in just about any form. If your smears smash on the way to us, we still work very hard to make new smears from the samples that you've sent us. The only time we can't read a smear is if it's got honey or wax on it. It interferes with our microscopy and our staining process, so we can't see anything through honey because we're looking at 1,000 times and through oil. It makes it very difficult for us, so we ask that we avoid as much honey or wax items on those. Up the right here is some honey. We also ask that honey is sent in as clean as possible. We understand that it's not always possible to get everything out of it, and that's fine, we can cope with that. But when it's got a lot of wax or comb and other materials in it, it interferes with that process when we're spinning the honey down, and we can't get a proper sample out of it to test. The other sample example here is just a pot that's just a bit too small. If you're going to send those in, that's fine. We just need 2 of them, but we need to know that they're the same sample. Otherwise, we will contact you and ask you the question, just in case that they're separate samples or anything like that. So I'd just like to say thank you to Michael Hornitsky, who is our consultant and bee guru here, and he's been a great source of information for me, and he supplied a lot of these images that I've used today. All my team at EMAI in the microbiology lab have been great support, and the management of EMAI. And we look forward to seeing all your bee, honey and slide samples, and we're here to help. So please contact us and let us know if there's anything we can do for you. Bye.